This lesson looks at cladistics. Here are the objectives. First, let's start by defining a clade. A clade is a group of organisms containing all of the descendants of a common ancestor. And it includes the common ancestor itself, as well as any descendants that are extinct. So a common ancestor and all of its descendants, existing and extinct, comprises a clade. Phylogeny means the evolutionary history of a species. And so cladistics is an approach to the classification of organisms, making use of this idea of a clade. The idea in cladistics is to organize all organisms into clades in order to reflect their evolutionary history. A major output of cladistics is called a cladogram, and a cladogram is just an evolutionary tree, also called a phylogenetic tree. Here's an example. Each branching point that we see here or here reflects a common ancestor. It also represents a speciation event. So if these are species A, B, and C, then this point here is the common ancestor of B and C, and it's also the speciation event whereby the lineage that gave rise to B and that gave rise to C split. Over here, this would be the common ancestor of A and B and C, and time is going in this direction. A tip for telling if a group on a cladogram is a clade is whether or not you could imagine cutting the tree at a single point and having the entire clade fall off. For example, here, if I were to snip here, B and C would fall off the tree, and that means B and C comprise a clade. If I were to cut here, that tells us that these species comprise a clade. Notice that A, B, and C do not comprise a clade because there's no place where I could cut the tree and only have A, B, and C fall off. So that's an easy way to tell from a cladogram whether a group of species comprise a clade. Now, different types of evidence can be used to build these evolutionary relationships, these cladograms or phylogenetic trees. Pretty much anything can be used. Morphological or structural evidence comparing how species look. You can use biochemical evidence or behavioral traits. Any traits that you can compare between different organisms. Also, DNA sequences or amino acid sequences can be used as well, and we'll talk more about those in a moment. The thing to be careful of is distinguishing homologous from analogous traits. Just to review, homologous traits are those that are similar due to common ancestry, whereas analogous traits are those that are similar due to convergent evolution. So, for example, the pentadactyl limb, the 5-digit limb, which has various different forms in different animals, is a homologous trait amongst those animals, similar because they inherited it from a common ancestor that had a 5-digit limb. Whereas the fins of fish and the fins of whales are an analogous trait because they arose not from a common ancestor, but because of convergent evolution. Now, for building these phylogenetic trees, we want to use homologous traits. And the difficulty can come in telling what's a homologous trait and what's an analogous trait. And so as a result of that, increasingly in recent years, cladistics has moved towards the use of DNA and amino acid sequences in order to tell relationships amongst organisms. The idea being that greater sequence similarity indicates closer relationships. So if you look at a particular stretch of the genome and B and C are very similar in that stretch in the sequence, and A is less similar, then that indicates that B and C are more closely related to each other than either is to A. In addition, some stretches of the genome and some stretches of amino acids and proteins actually evolve at roughly constant rates. And as a result of that, greater difference in sequence can tell you more than just relative relatedness, it can also tell you something about time. Greater difference in sequence implies greater time since divergence, and this can be calibrated in a way that you can figure out roughly how long ago species diverged. So here's a part of a tree. I'll call this A, B, and C again. And if we imagine that mutations are happening at a constant rate here over time in a particular gene, so each tick here represents a mutation, then we could look at the number of differences in the sequence between B and C. We'd have that many difference in sequences, and between these and A, and we'd have more. And so you could actually calibrate this. Instead of just being mutations, this scale could also tell you time. And that's a way of figuring out the time of divergence of different species and when different species evolved. And this concept and the DNA and amino acid sequences that can be used in this way, because they evolve at roughly constant rates, are called molecular clocks. So that's a quick look at cladistics, cladograms, and molecular clocks.